Chapter 12 Membrane Transport. Cells live and grow by exchanging molecules with their environment. The plasma membrane acts as a barrier that controls the transit of molecules into and out of the cell. Because the interior of the lipid bilayer is hydrophobic, as we saw in Chapter 11, the plasma membrane tends to block the passage of almost all water-soluble molecules. But various water-soluble molecules must be able to cross the plasma membrane. Cells must import nutrients such as sugars and amino acids, eliminate metabolic waste products such as CO2, and regulate the intracellular concentrations of a variety of inorganic ions. A few of these solutes, CO2 and O2 for example, can simply diffuse across the lipid bilayer, but the vast majority cannot. Instead, their transfer depends on specialized membrane transport proteins that span the lipid bilayer, providing private passageways across the membrane for select substances. Figure 12.1. In this chapter, we consider how membranes control the traffic of small molecules into and out of cells. Cells can also selectively transfer macromolecules such as proteins across their membranes, but this transport requires more elaborate machinery and is discussed in Chapter 15. Here, we begin by outlining some of the general principles that guide the passage of small, water-soluble molecules through cell membranes. We then examine, in turn, the two main classes of membrane proteins that mediate this transfer. A transporter, which has moving parts, can shift small molecules from one side of the membrane to the other by changing its shape. Solutes transported in this way can be either small organic molecules or inorganic ions. Channels, in contrast, form tiny hydrophilic pores in the membrane through which solutes can pass by diffusion. Most channels let through inorganic ions only and are therefore called ion channels. Because these ions are electrically charged, their movements can create powerful electric forces across the membrane. In the final part of the chapter, we discuss how these forces enable nerve cells to communicate, ultimately carrying out the astonishing range of behaviors of which the human brain is capable. Principles of membrane transport. To provide a foundation for discussing membrane transport, we first consider the differences in ion composition between a cell's interior and its environment. This will help make it clear why the transport of ions is both transporters and by both transporters and ion channels is of such fundamental importance to cells. The ion concentrations inside a cell are very different from those outside. Living cells maintain an internal ion composition that is very different from the ion composition in the fluid around them, and these differences are crucial for a cell's survival and function. Inorganic ions such as Na+, K+, Ca2+, Cl-, and H+ protons are the most plentiful of all the solutes in a cell's environment, and their movements across cell membranes play an essential part in many biological processes, including the activity of nerve cells, as we discuss later in this chapter, and the production of ATP by all cells, as we discuss in Chapter 14. Na+ is the most plentiful positively charged ion, cation, outside the cell, while K+ is the most plentiful inside. Table 12.1. For a cell to avoid being torn apart by electrical forces, the quantity of positive charge inside the cell must be balanced by an almost exactly equal quantity of negative charge there, and the same is true for the charge in the surrounding fluid. However, Tiny excesses of positive or negative charge concentrated in the neighborhood of the plasma membrane do occur, and they have important electrical effects, as we discuss later. The high concentration of Na+ outside the cell is balanced chiefly by extracellular CO-. The high concentration of K+ inside is balanced by a variety of negatively charged intracellular ions, anions. This differential distribution of ions inside and outside the cell is controlled in part by the activity of membrane transport proteins and in part by the permeability characteristics of the lipid bilayer itself. Lipid bilayers are impermeable to solutes and ions. The hydrophobic interior of the lipid bilayer creates a barrier to the passage of most hydrophilic molecules, including ions. They are as reluctant to enter a fatty environment as hydrophobic molecules are reluctant to enter water. But given enough time, virtually any molecule will diffuse across a lipid bilayer. The rate at which it diffuses, however, varies enormously depending on the size of the molecule and its solubility properties. In general, the smaller the molecule and the more soluble it is in oil, that is, the more hydrophobic or nonpolar it is, the more rapidly it will diffuse across. Thus, 1. Small nonpolar molecules such as molecular oxygen, O2, molecular mass 32 daltons, and carbon dioxide 44 daltons, readily dissolve in lipid bilayers and therefore rapidly diffuse across them. Indeed, cells require this permeability to gases for the cell respiration processes discussed in Chapter 14. 2. Uncharged polar molecules. Molecules with an uneven distribution of electrical charge also diffuse rapidly across a bilayer if they are small enough. Water, 18 daltons, and ethanol, 46 daltons, for example, cross fairly rapidly. Glycerol, 92 daltons, crosses less rapidly, and glucose, 180 daltons, crosses hardly at all. Figure 12.2. 3. In contrast, lipid bilayers are highly impermeable to all ions and charged molecules, no matter how small. The molecule's charge and their strong electrical attraction to water molecules inhibit them from entering the hydrocarbon phase of the bilayer. Thus, synthetic bilayers are 1,000,000,10⁹ times more permeable to water than they are to even such small ions as Na+ or K+. Cell membranes allow water and small nonpolar molecules to permeate by simple diffusion. But for cells to take up nutrients and release wastes, membranes must also allow the passage of many other molecules, such as ions, sugars, amino acids, nucleotides, and many cell metabolites. These molecules cross lipid bilayers far too slowly by simple diffusion. Thus, specialized membrane transport proteins are required to transfer them efficiently across cell membranes. Membrane transport proteins fall into two classes: transporters and channels. Membrane transport proteins occur in many forms and in all types of biological membranes. Each protein provides a private passageway across the membrane for a particular class of molecule, ions, sugars, or amino acids, for example. Most of these protein portals are even more exclusive, allowing entrance of only select members of a particular molecular class. Some, for example, are open to Na+ but not K+, others to K+ but not Na+. The set of membrane transport proteins present in the plasma membrane or in the membrane of an intracellular organelle determines exactly which solutes can pass into and out of that cell or organelle. Each type of membrane, therefore, has its own characteristic set of transport proteins. As discussed in Chapter 11, the membrane transport proteins that have been studied in detail have polypeptide chains that traverse the lipid bilayer multiple times. That is, they are multipass transmembrane proteins. See Figure 1124. By crisscrossing back and forth across the bilayer, the polypeptide chain forms a continuous protein-lined pathway that allows selected small hydrophilic molecules to cross the membrane without coming into direct contact with the hydrophobic interior of the lipid bilayer. Membrane transport proteins can be divided into two main classes: transporters and channels. The basic difference between transporters and channels is the way they discriminate between solutes, transporting some solutes but not others. Figure 12.3. Channels discriminate mainly on the basis of size and electric charge. If a channel is open, an ion or a molecule that is small enough and carries the appropriate charge can slip through, as through a narrow trapdoor. A transporter, on the other hand, allows passage only to those molecules or ions that fit into a binding site on the protein. It then transfers these molecules across the membrane one at a time by changing its own conformation, acting more like a turnstile than an open door. Transporters bind their solutes with great specificity in the same way that an enzyme binds its substrate, and it is this requirement for specific binding that makes the transport selective. Solutes cross membranes by passive or active transport. Transporters and channels allow small molecules to cross the cell membrane, but what controls whether these solutes move into the cell or out of it? In many cases, the direction of transport depends on the relative concentrations of the solute. Molecules will spontaneously flow downhill from a region of high concentration to a region of low concentration, provided a pathway exists. Such movements are called passive because they need no other driving force. If, for example, a solute is present at a higher concentration outside the cell than inside, and an appropriate channel or transporter is present in the plasma membrane, the solute will move spontaneously across the membrane down its concentration gradient into the cell by passive transport, sometimes called facilitated diffusion, without expenditure of energy by its membrane transport protein. All channels and many transporters act as conduits for such passive transport. To move a solute against its concentration gradient, however, a membrane transport protein must do work. It has to drive the flow uphill by coupling it to some other process that provides energy, as discussed in Chapter 3 for enzyme reactions. Transmembrane solute movement driven in this way is termed active transport, and it is carried out only by special types of transporters that can harness some energy source to the transport process. Figure 12.4. Because they drive the transport of solutes against their concentration gradient, many of these transporters are called pumps. We now examine a variety of transporters, both active and passive, and see how they function to move molecules across cell membranes. Transporters and their functions. Transporters are required for the movement of almost all small organic molecules across cell membranes, with the exception of fat-soluble molecules and small uncharged molecules that can pass directly through the lipid bilayer by simple diffusion. See Figure 12.2. Each transporter is highly selective, often transferring just one type of molecule, to guide and propel the complex traffic of small molecules into and out of the cell and between the cytosol and the different membrane-enclosed organelles. Each cellular membrane contains a set of different transporters appropriate to that particular membrane. For example, the plasma membrane contains transporters that import nutrients such as sugars, amino acids, and nucleotides. The lysosome membrane contains an H+ transporter that acidifies the lysosome interior, and the inner membrane of mitochondria contains transporters for importing the pyruvate that mitochondria use as fuel for generating ATP and for exporting ATP once it is synthesized. Figure 12.5. Although the detailed molecular mechanisms that underlie the movement of solutes are known for only a few transporters, the general principles that govern the function of these proteins are well understood. Concentration gradients and electrical forces drive passive transport. Solutes can cross the membrane by passive or active transport, and transporters are capable of facilitating both types of movement. See Figure 12.4. A simple example of a transporter that mediates passive transport is the glucose transporter, found in the plasma membrane of mammalian liver cells and many other cell types. The protein consists of a polypeptide chain that crosses the membrane at least 12 times. It is thought that the transporter can adopt at least 2 conformations and switches reversibly and randomly between them. In one conformation, the transporter exposes binding sites for glucose to the exterior of the cell. In the other, it exposes these sites to the interior of the cell. Figure 12.6. When sugar is plentiful outside a liver cell, as it is after a meal, glucose molecules bind to the transporter's externally displayed binding sites. When the protein switches conformation, it carries these molecules inward and releases them into the cytosol, where the glucose concentration is low. Conversely, when blood sugar levels are low, when you are hungry, the hormone glucagon stimulates the liver cell to produce large amounts of glucose by the breakdown of glycogen. As a result, the glucose concentration is higher inside the cell than outside, and glucose binds to any internally displayed binding sites on the transporter. When the protein switches conformation in the opposite direction, the glucose is transported out of the cell. The flow of glucose can thus go either way, according to the direction of the glucose concentration gradient across the membrane. Inward if glucose is more concentrated outside the cell than inside, and outward if the opposite is true. Transporters of this type, which permit a flux of solute but play no part in determining its direction, carry out passive transport. Although passive, the transport is highly selective. The binding sites in the glucose transporter bind only D-glucose and not, for example, its mirror image L-glucose, which the cell cannot use for glycolysis. For glucose, which is an uncharged molecule, the direction of passive transport is determined solely by its concentration gradient. For electrically charged molecules, either small organic ions or inorganic ions, an additional force comes into play. For reasons we explain later, most cell membranes have a voltage across them, a difference in the electrical potential on each side of the membrane, which is referred to as the membrane potential. This difference in potential exerts a force on any molecule that carries an electric charge. The cytoplasmic side of the plasma membrane is usually at a negative potential relative to the outside, and this tends to pull positively charged solutes into the cell and drive negatively charged ones out. At the same time, a charged solute will also tend to move down its concentration gradient. The net force driving a charged solute across the membrane is therefore a composite of two forces, one due to the concentration gradient and the other due to the voltage across the membrane. This net driving force is called the electrochemical gradient for the given solute. This gradient determines the direction of passive transport across the membrane. For some ions, the voltage and concentration gradient work in the same direction, creating a relatively steep electrochemical gradient. Figure 12.7a. This is the case for Na+, which is positively charged and at a higher concentration outside cells than inside. Na+, therefore tends to enter cells if given an opportunity. If, however, the voltage and concentration gradients have opposing effects, the resulting electrochemical gradient can be small. Figure 12.7b. This is the case for K+, a positively charged ion that is present at a much higher concentration inside cells than outside. Because of these opposing effects, K+ has a small electrochemical gradient across the membrane, despite its large concentration gradient, and therefore there is little net movement of K+ across the membrane. Active transport moves solutes against their electrochemical gradients. Of course, cells cannot rely solely on passive transport. Active transport of solutes against their electrochemical gradient is essential to maintain the intracellular ionic composition of cells and to import solutes that are at a lower concentration outside the cell than inside. Cells carry out active transport in three main ways. Figure 12.8. 1. Coupled transporters couple the uphill transport of one solute across the membrane to the downhill transport of another. 2. ATP-driven pumps couple uphill transport to the hydrolysis of ATP. 3. Light-driven pumps, which are found mainly in bacterial cells, couple uphill transport to an input of energy from light, as discussed for bacteriodopsin. See Figure 11.28. Because a substance has to be carried uphill before it can flow downhill, the different forms of active transport are necessarily linked. Thus, in the plasma membrane of an animal cell, an ATP-driven pump transports Na+ out of the cell against its electrochemical gradient, and this Na+ can then flow back in, down its electrochemical gradient. Because the ion flows through Na+ coupled transporters, the influx of Na+ provides an energy source that drives the active movement of many other substances into the cell against their electrochemical gradients. If the Na+ pump ceased operating, the Na+ gradient would soon run down, and transport through Na+ coupled transporters would come to a halt. The ATP-driven Na+ pump, therefore, has a central role in membrane transport in animal cells. In plant cells, fungi, and many bacteria, a similar role is played by ATP-driven H+ pumps that create an electrochemical gradient of H+ ions by pumping H+ out of the cell, as we discuss later. Animal cells use the energy of ATP hydrolysis to pump out Na+. The ATP-driven Na+ pump in animal cells hydrolyzes ATP to ADP to transport Na+ out of the cell. This pump is therefore not only a transporter but also an enzyme, an ATPase. At the same time, the protein couples the outward transport of Na+ to an inward transport of K+. The pump is therefore commonly known as the Na+K+ ATPase, or the Na+K+ pump. Figure 12.9. This transporter plays a central part in the energy economy of animal cells, typically accounting for 30% or more of their total ATP consumption. Like a bilge pump in a leaky ship, it operates ceaselessly to expel the Na+ that is constantly entering through other transporters and ion channels. In this way, the pump keeps the Na+ concentration in the cytosol about 10 to 30 times lower than in the extracellular fluid, and the K+ concentration about 10 to 30 times higher. See Table 12.1, page 388. Under normal conditions, the interior of most cells is at a negative electric potential compared with the exterior, so that positive ions tend to be pulled into the cell. This means that the inward electrochemical driving force for Na+ is large, as it includes the driving force due to the concentration gradient and a driving force in the same direction due to the voltage gradient. See Figure 12.7a. The Na+ outside the cell, on the uphill side of its electrochemical gradient, is like a large volume of water behind a high dam. It represents a very large store of energy. Figure 12.10. Even if one artificially halts the operation of the Na+K+ pump with a toxin such as the plant glycoside ouabain, the energy in this store is sufficient to sustain for many minutes the other transport processes that are driven by the downhill flow of Na+. For K+, the situation is different. The electrical force is the same as for Na+, because it depends only on the charge carried by the ion. The concentration gradient, however, is in the opposite direction. The result, under normal conditions, is that the net driving force for movement of K+ across the membrane is close to zero. The electrical force pulling K+ into the cell is almost exactly balanced by the concentration gradient tending to drive it out.